So in this chapter, we start talking about the renin-angiotensin-aldosterone system. It is really important, and I believe you will have to study this again in your physiology class. So as we know, renin is secreted from the kidneys, from the granular cells, which are present in the afferent arterioles. Where do we get angiotensinogen? It is from the liver. Where do we get aldosterone? They are from the adrenal glands. Yeah, zona glomerulosa, layer, adrenal cortex. So I want you to be clear of these renin-angiotensinogen, which will become angiotensin, and later we go to the adrenal gland to secrete aldosterone. So this is a very good diagram. I got it from Wikipedia. So our kidneys from the granular cells of the afferent arteriole, we secrete renin. That renin will convert angiotensinogen, which is secreted from the kidney, into angiotensin 1. And in the lungs, surface of the pulmonary and renal endothelium, we have the angiotensin converting enzyme, which we call ACE, angiotensin converting enzyme. Angiotensin converting enzyme will convert angiotensin 1 into angiotensin 2. Angiotensin 2 will trigger many physiological effects. It will increase the sympathetic activities. It will stimulate the tubular sodium and chloride reabsorption, and in exchange of potassium, and there will be water retention. And it will go to the adrenal gland to stimulate the adrenal cortex for aldosterone. It will increase the vasospasm in the arteriole to increase the blood pressure. And it will go to the pituitary gland to secrete antidiuretic hormone, ADH, which will comes down to the collecting duct to increase the water resorption from the collecting duct. So angiotensin 2 has many physiologic effects. We got angiotensin 2 from angiotensin 1, and the enzyme ACE changed that 1 to 2. And therefore, I believe we have heard about many ACE inhibitors, angiotensin converting enzyme inhibitors, used in our medical practice. So here we are. Now let's talk about ureters. These are the tubes which carries urine from the kidney into the urinary bladder. They are about 10 inches long. They are running at the tip of the transverse processes of lumbar 2 to lumbar 5, and they are crossing in front of the sacroiliac joints and enters into the bladder obliquely. So this is the outline of the ureter, and these are the shadows of the kidneys and renal pelvis. You can see the major and minor calyces here, and the urine is formed in the kidney and runs along the ureter, which is running on the tip of the lumbar vertebral transverse process, lumbar 2, 3, 4, 5, and goes in front of the sacroiliac joint and enters into the urinary bladder. And if we do a cross section and look under the microscope, histology of the ureter, we will see the mucosa layer, mast cell layer, and outer connective tissue adventitia layer. In the mucosa, it is made of transitional epithelium and mast cell layer, inner circular, and as longitudinal and circular layer, and outer connective tissue adventitia layer. Let's talk about urinary bladder. If you have been to the anatomy dissection lab, I believe you have seen the urinary bladder, which is just lying posterior to the pubic symphysis. The arteries for the bladder comes from the internal iliac artery, which are called superior and inferior vesical arteries. Vesica means bladder. A bladder is an extraperitoneal organ. It is like a triangular or pyramidal shape, which is upside down, and ureters enter into the bladder posterolaterally. Posterior wall on the inferior surface of the bladder is called trigone of the bladder, and average capacity is about 500 ml, and the neck of the bladder continue as the urethra. So now this is the diagram which is explaining the allantoids and these allantoids will become the feeder uric acid, URACHUS, feeder uric acid, and that uric acid connects the fetus urinary bladder to the yolk sac. So I'm just trying to say all these things because at some point we have to talk about the embryology. Like as we know, like bladder has 3 layers from inside out. The epithelial lining of the bladder is transitional epithelium, which sits on the lamina propria. The mast cell is called detrusor mast cell, which is very thick and it is expandable. And the outermost is connective tissue adventitia. And if you look at the histology of the bladder, these are the walls of the bladder, and you can see the innermost, which is in contact with the urine. This epithelium is the transitional epithelium. And finally, we have the very thick mast cell, detrusor mast cell layer. And outermost is the adventitia connective tissue. Blood vessel wall, as we know, we have on 3 layers: tunica, intima, media, adventitia. Urinary bladder, we have 3 layers. And the intestinal layers, we have to study 4 layers: mucosa, submucosa, muscularis externa, and serosa. So this is what we say about the trigone, which is the in the bladder, triangle of the bladder, as you can see here. And you can see the ureters from the right kidney, ureters from the left kidneys enter into the bladder in the posterior and lateral wall here. And this triangular shape is the triangle of the bladder. And bladder inner lining of the bladder is transitional epithelium. It has a very thick mast cell layer. This mast cell is called detrusor mast cell, and outer adventitia layer. And our ureter, we leave the our urinary bladder, we continue and come out as the urethra. And here we have the difference between male and female. Now this is the male urinary bladder, and this is the female urinary bladder. And female urinary bladder can continue as urethra, URETHRA, which is very short and finally ends as the external urethral orifice here. And here for the male urethra, it has to go inside the prostate, which we call prostatic portion of the urethra. It has to go through the external urethral sphincter, which is what we call urogenital diaphragm. And there we call the intermediate part of the or muscular part of the urethra. And finally, which is inside the penis, and that is what we call penile portion of the urethra. So male urethra is much longer comparing to the female urethra, and therefore females are more prone to urinary tract infection because of their proximity of the external urethral orifice to the urinary bladder. And this is the lateral view, sagittal view. And now we can see the urinary bladder, which comes out as the urethra in the prostate, therefore prostatic urethra. And now it is crossing the urogenital diaphragm. It is the membranous urethra and the spawn going in the penis and external genitalia as the penile urethra. And here we can see the structures coming into the seminal vesicles and ejaculatory ducts coming into the prostatic portion of the urethra. And in female, as we have seen, the route is much shorter, with the bladder go through the urogenital diaphragm and come out as the external urethral meatus. So urethra is also lined by the transitional or might be pseudostratified columnar or stratified squamous epithelia. So it varies. It has internal urethral sphincter, which is made of detrusor muscle, which is involuntary, external urethral sphincter, which is made of urogenital diaphragm, which is the skeletal muscle. In female, it is much shorter. We saw that. It is just about 1.5 inches. In male, it is about 8 inches. And the path it has to take is through the prostate. Therefore, we call it prostatic urethra, through the urogenital diaphragm. Therefore, we call it membranous urethra, and the penile urethra or the spongy urethra. Of course, our urethra will end as the external urethral meatus, and I believe you will be able to understand this diagram. So now let's go through of the micturition. As we know that our urinary bladder capacity is 500 ml. So when there is, let's say about 500 ml of urine in the bladder, our bladder get distended, and this way be taken to the central nervous system with the pelvic nerves, as you can see here, go up along the spinal cord and go to the pontine micturition center, and the reflexes come back down, and on one way it will, with the pelvic splenic nerve, it will contract the urinary bladder. On the other hand, it will contract the external urethral sphincter if the situation is not appropriate. And at the same time, these descending fibers come along the inferior hypogastric ganglia. It will stimulate the urinary bladder. So that is the micturition process. The voluntary control is external urethral sphincter, which is the urogenital diaphragm. And parasympathetic, we stimulate the contraction of the detrusor muscle. Sympathetic, we inhibit the detrusor muscles. Let's talk a little bit about the development of the kidneys and urogenital ridges. So during our embryonic period, before we get the kidneys, we have a small little buds, which we call pronephros. Nephros mean kidney, pro mean pre, but they degenerate and disintegrate. And later, we have another structure, which is called mesonephros. They degenerate as well. And finally, we have metanephros. These metanephros will develop into kidneys. Urogenital sinus will become the urinary bladder and urethra. And allantoids, that's what we saw in the previous diagram, we form into urachus of the bladder. The kidneys, originally they were formed in the pelvic region with the gradual development of the fetus, and their position gradually move upwards into the lumbar region. And therefore, we mention like ascending of the kidneys. So from the pelvic portion, go up into the lumbar region, and we call it ascent of the kidneys. Week 5, week 6, here, that's what we are forming, like pronephros, mesonephros here, mesonephros here, which we disintegrate, pronephros, which we degenerate as well, and finally the metanephros here, as you can see, which form pretty low down in the lower part of the fetus with the gradual development of the lower part, and this makes the metanephros kidney to move upward into the lumbar region. And here now you can see the metanephros kidney, which is gradually moving as the lower part portion is gradually longer and longer. And now, if we are not lucky enough, our two kidneys, they stay fused in the inferior pole and they cannot go upwards. That is the variance of the kidney. Therefore, you might heard about the pelvic kidneys or the kidneys which cannot ascend up to the lumbar region. And now here we have done this exercise in the lab as well. We are looking at the renal corpuscle, and these are the cross-section, which might be proximal convoluted tubule, distal convoluted tubules, and they are lined by the cuboidal epithelium, as you can see here. And now this is the outline of the ureter, which is running on the tip of the transverse processes of the lumbar 2, lumbar 3, lumbar 4, and lumbar 5, and it goes into the urinary bladder. And now this is the colored CT, this shows the right kidney, and this shows the left kidney. And here we have multiple cysts, which are the fluid-filled sac here in the kidney. And of course, we see the multiple cysts in the kidney, and therefore we call it polycystic kidneys. And now we can see the CT angiogram, which is showing the abdominal aorta, which is abnormally bulged out here, which we call aneurysm. Yes, this abdominal aorta dividing into right and left common iliac arteries dividing into internal iliac and external iliac arteries. And you can see the renal arteries here, which is going to the kidneys. And you can see the here, renal arteries here. I believe this must be the superior mesenteric artery, which is A, because it is above the level of the renal artery, which is impaired. And now this X-ray show the little radio-opaque structure, which we will say is a stone, and it is in the outline of the ureter, and I believe this stone is stuck in the ureter. This is the end of the urinary system lecture, and I hope you will go back and study again. Thank you for your attention.